Fab. So we'll get started. So today we're going to cover some respiratory embryology. I've tried to make it kind of as condensed and as simple as possible because, as you guys probably know, it is very complex and confusing, and you can honestly spend like all day online looking at various things. So I've tried to break it down and make it quite simple, and just get the key information. So in the session, we're going to cover the formation of the lung buds, lung maturation, and the production of surfactant, and we're going to look at the respiratory changes at birth as well. So a quick recap from my last embryology session, where we looked at the start of the embryo folding. So as you guys probably know, we end up with a trilaminar disc, which is made up of the endoderm, ectoderm, and mesoderm. And does anyone know when this folding, at what week this starts to happen? It's okay if not, I'm just going to see if anyone knows. No? So it's the start of week 4 that we start to have this folding. So you can see in these diagrams here, we actually have longitudinal folding down the center, so from the head to the tail, and we have transverse folding or lateral flexion as well across the middle. And this leads to the formation of the abdominal wall and all your internal organs pretty much. So like it says here, the folding process encloses the mesoderm and the endoderm, and then we have the ectoderm on the outside, which actually becomes the skin. So that's just a quick recap from last session. This shows it in a bit more detail. So you can see here we have the amniotic cavity. This is still in week 4. And as it's folding laterally, so it's folding round the sides, it begins to pinch off this yolk sac at the bottom that we can see in yellow here. Does anyone know the purpose of the yolk sac at all? Oh, apologies. It's okay if not. So the purpose of the yolk sac is actually to provide like nutrients and gas exchange between the mother and the embryo before the placenta takes over. So that's still in week 3 and week 4. This is at very early stages. So you can see here during this pinching that we have this intraembryonic coelom, I think it's pronounced. There's 2 of them at the moment that are separated, and these actually give rise to the thoracic and abdominal cavities. And then we have this somatopleura down here where you can see labeled, which is where the mesoderm and the ectoderm come into contact and they develop the internal body wall. So another nice diagram that we can see here. So in pale, we've got the amniotic cavity around the edge, and then we can see the skin ectoderm in pale blue. You can see here now that we've got these intraembryonic coeloms, which then fuse together now instead of being 2 separate ones to form like a horse shape. A horseshoe shape even. The main part of this diagram that I want you to take away is that we've got this, you can see it ringed in yellow, this embryonic gut tube. So that is actually endoderm in yellow, surrounded by the mesoderm in the red. And the embryonic gut tube is what actually gives rise to the respiratory system or the respiratory bud. You can also see we've got the splanchnic mesoderm, which gives rise to connective tissue of the GI tract, smooth muscle and blood vessels. And we've got the somatic mesoderm as well, which contributes to connective tissue of the body and the limbs. Okay, so formation of the lung buds. I've tried to make it as simplistic as possible. This is in a stepwise pattern. So you can see here, we've got this endoderm in this pale yellow color, and it's surrounded or coated in this splanchnic mesoderm. And we've got a primordial pharynx. So it's like in the very early stages. And you can see coming off that, we've got a laryngotracheal opening, and then like a diverticulum, which is just, as you know, like a kind of thing coming off the side. And this appears on the ventral wall of the primitive foregut endoderm, posterior to the primordial or primitive pharynx. And then we have this further development of this diverticulum into this respiratory bud. So this is now in the very, very early stages of forming lung buds. And then you can see as we go along, this has now properly become a laryngotracheal tube, And we've got the formation of a left and right primary bronchial buds. So they're forming the basis of your left and right bronchial tree at this point. And also during this development, we've got a tracheal esophageal septum. So as you can see at the bottom here, this further develops and pinches off these two structures. So I think this is at the end of week 5 at this point. We now have a completely separate esophagus, which you can kind of see on the left side. And we have a trachea on the right side on our primary bronchioles coming off it. So this shows it quite well. This is kind of just repeating what I've said. So you can see at day 28, we've got this primitive trachea and we've got a left and right bronchial bud. By day 35, we've now got secondary bronchi forming. This further develops at day 42. And then by day 56, we've got a pretty good bronchial tree at this point that's formed. And again, over on the right-hand side, so you can see at 5 weeks, we've got this laryngotracheal fold and groove coming off with the esophagus posterior. We've got our left and right primary bronchi or lung buds covered in visceral mesoderm. So if you guys can remember, we have visceral mesoderm going round very close to the organ edge, and then we've got parietal mesoderm as well. And then you can see 1 week later, we've got branching now. So I kind of think of it as like a tree formation. We'll have secondary and tertiary bronchi branching off as well. This is just a small thing. I think you guys just need to be aware of it, not in too much detail, but we have something called a pericardio-peritoneal canal. So this is simplistically just the space where the lung buds will grow into to become the adult fully developed lungs. You can see here, if you can see these kind of where the blue arrows are, these two kind of indents, this is where the lungs will sit in. And we have you can see the lung bud forming there as well. This kind of pinky purpley structure, the septum transversum, this actually gives rise to the thoracic diaphragm and the ventral mesentery of the foregut. This septum transversum just forms from a very thick sheet of mesoderm, and it's rostral to the developing heart. So I think just to be aware of that. So surfactant production. Does anyone know the actual purpose of surfactant and why we actually need it? No? Okay, so we need surfactant to reduce surface tension at the air-water interface of the alveoli, and it prevents them collapsing. So as you can see on this nice diagram on the right, the lumen of the alveoli aligned with molecules of water, and the water actually is what carries the surface tension, as there's intermolecular hydrogen bonding between water molecules. And this surface tension will cause the alveoli to collapse in on themselves. And then obviously you have like an impossibly small surface area, so no gas exchange can occur. So we have surfactant secretant from type 2 alveolar cells, which secrete the pulmonary surfactant, and that accumulates in the lumen of the alveoli. And this prevents the high degree of surface tension that would otherwise happen, and it prevents them from collapsing, and therefore we can have proper gas exchange. Again, this is quite simplistic. It's just a repeat of what I've really said earlier. So we have continual development of this bronchial tree. So we start off with our our left main bronchus and right main bronchus. Then we go to our secondary bronchus, our tertiary bronchus, a terminal bronchus, as you can see down here. And at this point, all these structures have plates or rings of cartilage, which you can see. So these blue bands almost are cartilage, and this keeps the bronchi very rigid and stops them collapsing basically. When we get down, so past the terminal bronchus into the bronchioles, and then you can actually see on the left image again now, we've got the terminal bronchioles, then these branch off into respiratory bronchioles, and then we have the alveoli here. So we have the alveoli ducts and the sacs, And these are composed of smooth muscle, so there's no plates of cartilage past this point. Another important thing to discuss is pleura. So I have touched on it briefly. The endoderm tube is still covered in splanchnic mesoderm, and the splanchnic mesoderm basically becomes the visceral pleura, and the somatic mesoderm becomes the parietal pleura. So the visceral pleura is, as you can see on the right, is what surrounds the bronchial tree. It's in very, very close contact with it. And then we have our parietal pleura that's more on the outside of the thoracic, within the thoracic cavity, but surrounding other structures as well. Again, this is just a repeat of what I said, so we can see the time scale of things. So day 28, we've got our budding of our bronchial buds. It's quite important, I think, to remember that that happens at day 28 or 4 weeks. Then knowing that we have subsequent development on a weekly basis, it's quite quick, really, up to day 56, where you can see now we've got pretty well formed 3 lobes and 2 lobes on the left even, and you can see our primary, secondary and tertiary bronchi that are formed. So there's 4 stages that you need to be aware of in lung development in the embryo. So we'll go through them now. They've all got slightly awkward names. So the first stage is a pseudoglandular stage, which happens at 6 to 16 weeks. At this stage, the lung tissue of an embryo can be very similar to an exocrine gland if you were to look at it down a microscope. So you can see on the right here, we can see lots of connective tissue cells, capillaries that are kind of floating around, and a terminal bronchiole, but there's not really much going on in terms of any interaction between the two. All major elements have formed apart from respiratory regions, so it's quite simplistic at this point. The second stage is a canalicular stage, I think that's how you pronounce it, at weeks 16 to 26. So we've had quite a development at this point. The tissue has become very vascularized. The lumen of the bronchi and the terminal bronchi have grown in size. Each terminal bronchus, so if you remember, that's the very last stage before the respiratory bronchi, they now have at least 2 respiratory bronchi branching from them. And we have a thin wall within these terminal sacs of the alveoli now. And that's really important to allow an interaction to commence now between the surrounding connective tissue. So now we have the saccular or the terminal sac stage, and this happens from 26 weeks till birth now. So we have even more terminal sacs developing with our alveoli. We have our type 2 pneumocytes or alveolar cells form, and this is what, like I've said, secretes surfactant. And the epithelia becomes very thin within these terminal sacs. And now you can see an interaction between this very thin epithelia and the surrounding capillaries in the connective tissue. You can also see we've got some involvement of some smooth muscle, some fibroblasts as well. And the fourth and final stage is the alveolar stage, which happens from 32 weeks until 8 years old. So remember that this happens after birth primarily. So yeah, 95% of alveoli develop postnatally. At birth, there's just primordial or very early stage alveoli developed, and they're simply small bulges coming off the respiratory bronchioles and the alveolar sacs. Important to note that there is a size increase in the lungs postnatally, but this isn't actually due to the lung tissue itself. It's due to an increased number of these respiratory bronchioles and alveoli forming. Yeah, so the fourth stage needs to be aware of. Respiratory changes at birth is really important. So we have an initial fetal breathing movements, which obviously occur within the mother, and it's breathing like movements that occur episodically or often quite randomly in the fetus, and it's usually due to like a contraction of their diaphragm. Obviously the fetus is not able to breathe, their lungs are still in fluid, the whole purpose of this is to start to exercise their accessory respiratory muscles to kind of strengthen them ready to breathe when they are born. So at birth, there is aspiration of this amniotic fluid that's within the lungs, and this is cleared via 3 routes. So predominantly it's from the nose and the mouth. When the baby's born, this can either be from their first breath that they naturally take, or on delivery from the vaginal wall pressure during labour. It pushes it out. The amniotic fluid can also be absorbed into the pulmonary circulation or into the pulmonary lymphatic system as well. It's also important to be aware of cardiovascular changes that occur when the baby's born. So as you can see on this diagram, it's a bit complex, but the 2 main kind of holes you need to be aware of are the foramen ovale, which we have between the left and the right atria, and the ductus arteriosus, which is between the aortic arch and the pulmonary artery. So these are both open when in the embryo before it's born, and it allows the bypassage of blood through the lungs. So obviously they're not using their lungs when they're still in the mother, and it allows this blood to be transferred from either side of the heart and bypass the lungs, and then this oxygenated blood can then be pumped up into the aortic arch, and it can go down the abdominal aorta into the iliac arteries and so on to the limbs. Yeah, I will go back. So another important thing to know that you'll do in your cardiovascular system is these 2 holes close at birth, but they can obviously stay open in some children. Yeah, so neonatal respiratory distress syndrome. I thought I put in a little clinical thing in here as it's quite interesting. It's due to insufficient surfactant production or immature lung development. So they have a low number of alveoli at birth. It usually occurs in neonates under 32 weeks old. So you can see an occurrence, it's only 1% in full-term babies, but it can be up to 50% in preterm neonates. And it is simply due to they haven't had really enough time to develop their lungs properly, and they've not got really any type 2 pneumocytes to produce a surfactant. This can obviously have huge implications. Like I've said before, we need the surfactant to keep the alveoli open and stop them collapsing. So you can see down here that they can present with cyanosis, tachycardia, tachypnea, they're struggling to breathe, nasal flaring, they're trying to get more air inside of them, and so on, chest wall recessions as well. Prenatal treatment with glucocorticoids stimulates surfactant production. So this can actually be picked up within the womb and can be treated actually before birth sometimes. Okay, so that's everything. I hope that's given a quick run through. Here's some additional useful resources I like to use. So the first one's a really good embryology website. The second 2 are YouTube channels that I think are really good for embryology as well. Thank you for coming. I hope that was a good whistle-stop tour. I do have a feedback form on here, the link's there if you guys would be so kind as to fill it in for me, would be wonderful. And that is it. Thank you.